Hello again guys, I'm back. And for this video, we will talk about DNA transcription.
DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with the promoter and ends with the terminator. Genes can also contain regulatory sequences that can be found near the promoter or at more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In this video, we will talk about transcription. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then, during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding sections must be removed and modifications such as 5' cap and a poly-A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus and ready to be used in translation.